Hey everybody, it's Dr. Phil from medpathway.com. We're going to look at the skinny on making fat. How do you make fat? And so we know you make fat from excess glucose, but I also included fructose because we all talk about fructose corn syrup. And we know that glucose is a precursor to make fat, but it's also a precursor to make glycogen as well. And I haven't shown you the pentose phosphate pathway. We've kept that out for simplicity. But what you see is glucokinase and fructokinase. And so that's a clue that this process has occurred in the liver. Now, most fat's made in the liver. Not a lot of tissues want to make fat. They want to burn fat for energy, but nobody really wants the burden of making it. Intestines make it, and so do adipose tissue, but this process occurs in the liver when you have high insulin and low glucagon. And so think of high insulin as something that's secreted after you eat a giant carbohydrate meal. So the glucose is going to be oxidized into pyruvate via glycolysis, and what you see is that this is expedited by insulin. We also see that glycogen synthesis occurs with high insulin as well. And so glycolysis is the conversion of glucose into pyruvate, and then what happens to the pyruvate? It's shuttled into the mitochondria where the PDH, or the pyruvate dehydrogenase complex, can oxidize those three carbons into a two-carbon entity called acetylCoA. And we all know that acetylCoA combines with oxaloacetate, or OAA, to generate citrate, which is the first step in the TCA cycle. Now, most of the time you think of the TCA cycle as having the capacity in the mitochondria to oxidize carbon into CO2. And yes, that occurs, but what if you ate so much sugar and made so much pyruvate acetylCoA that the enzyme isocitrate dehydrogenase couldn't really handle all that citrate? What would happen? Well, what would happen is that you would slow the TCA cycle down. The citrate would build up because the isocitrate is at its Vmax, but you keep giving it more citrate. So it can't handle the citrate. It can't oxidize all of it. So some of it builds up, achieves a certain concentration, and then it oozes out of the mitochondria into the cytosol, where you actually serve as a substrate for making fat by the fatty acid synthase and some additional enzymes that are not shown. So fatty acid synthesis occurs in the cytosol from citrate, but the citrate's initially derived in the mitochondria. Come check us out from medpathway.com.